My name is Marc Caporini. I am a director in research systems innovation at Moderna. And I'm going to talk to you guys about an automation project for research that we've been working on for the past year and a half, 2 years. Really around building a data generation engine for powering AI using automation and robotics. So one other thing I've noticed, this is not the crowd to interrupt and ask questions, but I would much prefer this to be a conversation rather than a talk track. So if you do have questions, please raise your hand, grab the mic. I would rather have a back and forth, especially when I'm doing some of the demo things in the middle. So thank you. All right, so just a quick outline of what I'm going to run through today. I'd like to start with sort of how do we create value for patients? I'll go to how research creates value through data and know-how. Then I'll move on to how I think automation for research really supports that goal, and how automating for flexibility and changing our minds is really that key piece when we're researching something new. Then I have a sort of a data exploration demo from some data from our pilot system. I'll show you how we built flexibility through really a hyper focus on data and not information, so make a distinction between data and information. And then I have some examples of the rapid changes we made while building both the pilot system and what we call our V0 system, which is now sort of arrived and we're standing up right now. And lastly, I want to highlight some tools that enable scientists to build and drive cultural change. And artificial is one of those, and there's a few others that you'll see along the way. To me, this is a big key to getting adoption and really getting those tools that empower the scientists to build and use these systems. All right, so what drives value for patients? I sort of try to start at a very high level. It's the real value driving question is, how do we have access to a diverse set of safe and effective medicines at a low cost and right now? So in order to do that, how do we get medicines? We need approvals. And so how do we get approvals? We need data packages that support, we know what we're making and we're making it consistently. So how do we get those data packages? We need clinical trials to show that it works. So how do we get to a clinical trial? We need a data package from research that moves us. So everything that moves us through the pipeline is really driven around data. And I would say the only valuable thing that we create in research is that data and that know-how. We don't ship product in the research. All the materials we make, they're only there to create the data. So it's really all about capturing that data. And then for automation, I think about increasing both the throughput and reducing the cycle time. So every time we take something forward and fail, we basically go back and start over again. And that cycle is expensive and it takes time, and that's time that we're not getting medicines to patients. So really that's the driver for us. So what are we aiming to do here? We're aiming to think about the drug development ecosystem and create an infrastructure that allows that process to be much faster than it currently is today. So I'm going to kind of work us around this cycle here. So in our space, when we're thinking about the drugs that we're developing, we're really starting with the chemical component. And so we take different chemicals, we're exploring a lot of different chemical space, and then we put those together into a sort of candidate for a drug product. And then we basically measure a bunch of biophysical data. So for LNPs, we're measuring things like size, we're measuring encapsulation, like how much RNA is inside there. And then we usually have some process data, basically how do we make these LNPs, how do we stitch together the chemicals in a certain way. And then we measure some in vitro biology, usually cell-based assays, we're looking for a response. And then finally we go to in vivo. And so that's sort of the DMTA cycle that we sort of work through here and how we sort of think about these things. So we want to accelerate that cycle. And then on the right side, we're kind of thinking about it's automation is more than just the physical tools. Getting a liquid handler is not going to get us there. Is we really need to think about automating the data and orchestrating the data as well. So my team in particular, we sort of sit this interface of a bench scientist, digital transformation, and operational excellence. And we really try to do all of those together and thinking about not just how do we do what we're currently doing today better, but what should we be doing in the future and how should we be doing it, and sort of mixing all three of those components together. Okay, so making an LNP or LNP medicines, it's a lot like cooking. I know this analogy is used a lot. You have your input chemistry, which is really your components. It's like your ingredients. Then you have a recipe, so you have certain combinations that you mix them together, and then you have sort of your utensils and your appliances, basically like machinery used to make things. And I think what a lot of traditional high throughput screening approaches, what they look like is really, they have a very limited set of recipes and a limited set of utensils, and they run tons of ingredients through there. And if you're running a Michelin 5-star restaurant, you probably don't want to use the same recipe and the same process for every single one of your dishes and just change the ingredients. Like, that's not going to work. We really need to explore the flexibility across that full space. And so this is where I think we've really tried to shift our philosophy from like a traditional automation approach, much more linear thinking, looking like, we're making 1,000 things a day the same way every day. And so what we're trying to build is something that looks more like a network process. So you have this modularity, you have these pieces that fit together in a network, and you're constantly changing that network to optimize for a different process. So really we want to explore like thousands of different processes or thousands of different recipes a few times each, or like 10 or dozens of times. So that's a very different automation system. It looks and feels very different than, I think, a traditional one. And so this is sort of a slide to lay out some of our goals and our philosophy. So what we're trying to do here is not a fully automated approach like AI in the loop doing everything, but really a hybrid approach. And so we want this augmented end-to-end ecosystem where we can increase the throughput and we can reduce the hands-on time that scientists are doing, so they can spend more time in the analyze and sort of design phases, places where you need creativity, rather than, say, pipetting or doing measurements, or the worst case scenario, which is like cutting and pasting from Excel. So that's like, I don't want them doing that type of manual data analysis. And the other important thing where we are now in this development is, I think it's very important to have the scientists in the loop and have them be bought in. So it's really like giving them tools to leverage their accumulated knowledge and their ideas, but put those to work in a very rapid cycle time way. And so in doing this, we really want to get like a lot of different perspectives on this. We want to expand the space. We're going to reduce the development time. All of these things get the speed to market. And I think this is the approach that is going to get us there. And so, again, thinking about the philosophy of how we went about building this, is really probably spent more time talking about the philosophy than actually building it. But we started with the application and purpose first, and then we asked the question of like, what data do we need, and what software can get us that data, and then finally we get to the hardware and the tooling. So there's kind of a different approach, but we set out to do this and then looked at all of these different tools from digital tools. We worked with different vendors and sort of vetted them on certain things, hardware, and then finally integrator selection. But one of the key things we did was, at each step we were asking them, like, what is the data we need out of this, and how are we going to connect it to all of the other data pieces? And we were actually, when we were vetting vendors, we spent a lot of time really looking for that philosophical alignment. It's like, do they get it? Like, do they understand that, like, I need an API that gets me the data the way I want it, or allows me to drive an instrument the way I want to do it, not necessarily the fanciest piece of hardware, the newest piece of hardware. So that was a very different approach for working with some of the vendors, and it led us to some interesting choices and sort of where we are now. I will say we kicked this off about 2 years ago. The landscape has changed so much that if we went and did it again today, I'm not sure we would have selected exactly the same vendors. Like, the vendors have moved quite a bit in that time. Okay, so I'm going to start to dive a little bit into the specifics of the science and sort of LNP engineering. So lipid nanoparticles for delivering mRNA are kind of unique. I think they're quite different, especially from like a small molecule approach in pharma. So you have these components. You have an ionizable lipid, usually a sterol, a phospholipid, a PEG lipid, and all of those sort of go around and encapsulate the RNA. Now you can change the chemicals you're using, you can change the compositions you're using, and you can change the process. All of those things will result in, say, a different structured LNP, a different heterogeneity of those LNPs, and there's many different things that we can measure biophysically about them. And then there's a whole, beyond that, there's effects on chemical stability and physical stability. Do the LNPs hold up over time? Then you have different routes of emission. So depending on how you're entering the body and what cells you're targeting, you may want to design your LNP to deliver differently. And then you get into the fun biology part, which is the LNP has to get into the cell, has to get into the cell that you want it to go into and not other cells. And then finally, you have to get the RNA released into the cytosol and translated by the ribosome into the protein of interest. There's many, many different pathways to get there. And traditionally, if you're thinking about a small molecule drug, you kind of know the rules. They've been around a while, and you can basically ignore a lot of the interactions and think only about the small molecule. Here, we don't necessarily know all the rules, so it's very hard to down select along the way and understand if we're actually sampling the space broadly enough. And so I'll try to expand on that a little bit in this diagram. So if you have all these different components, they individually might have some effects on the things we measure: size, polydispersity, stability, expression, distribution in the body. But each one, they're kind of coupled to the other. So it's not just like a one-to-one first order effect. It's really the effects in combination that kind of overwhelm the first order effects, and you have to sort of treat it as like one complex system. And I think there's a certain irreducible complexity about these systems that I personally like at the moment. I think eventually we will get to rules to reduce them, but at this point, like, it's really important to just embrace that complexity and try to get data to then reduce it. And so along those lines, how have scientists traditionally dealt with these high dimensionality spaces? And that is, we sort of make some assumptions and we start testing them. So we come up with a hypothesis, and we think it behaves this way. And so we explore this space, maybe in this multidimensional space over here, and then we get another idea, and we come up with another hypothesis and do another design, make, test, analyze cycle, and okay, maybe that explores this space over here. And we might generalize from that and pick another hypothesis and say maybe that explores here. The problem is, when you do these individual explorations, I think most scientists and more importantly, most leaders want to overgeneralize the results of and conclusions from those spaces. But if those spaces don't overlap, it's like, how do you know really what generalizes to the large space? So this has been one of the drivers for us, and not just knowing what space we've explored, but understanding what spaces we haven't explored. And I have some anecdotes I can share on that a little bit later as well. So I'm going to kind of lay out this input space and sort of the how we make things, how we measure things in the output space for a small demo set that I'm going to show. So on the left, I have what I call the input. These are the things we can control. And we start with like chemistry. So we can pick the molecules that we want to test, and then we can mix them in different ratios. So that's sort of our composition. And then we have process. So the order of mixing and the way we actually mix them. So you have 6 different ingredients and you mix them in different ways, you can actually get different performance based on how you mix them. So you can't really simplify it to just chemistry and composition. And so even though I'm showing that as like 3-dimensional plot, it's actually much higher dimension than that. Each one of those things is sort of its own dimension. And then on the right, I have this output space. So again, I'm drastically oversimplifying here. I split it just in 2 of biophysical measurements, so size, encapsulation, concentration, things like that, and then biological measurements. Here, say multiple cell lines, multiple readouts of expression or uptake. You can also go to in vivo as well. And so for this particular set that I'm going to show, I call it a small demo because it is much smaller than where we're going. We looked at 6 different lipids with over 200 different unique compositions. This is just like a 4-component composition. You can easily go to 5 or more components, and just one process in this particular case. So that's like a 9-dimensional space. Could be much larger. And for the output space, I'm going to limit it just for this demo of, we're going to look at size and encapsulation, and then I have expression from 2 different cell lines. And so if you give me just a moment, I am actually going to switch to dashboard. This is amazing. I'm actually going to do a live demo here. Okay, so this is a blinded dashboard for some of the data that we've collected. My dream is to stop doing PowerPoint decks for presentations and just go to the dashboard. We've done this a couple of times for other projects. It works really well. People can ask questions and you can answer them on the fly. So I'm just going to quickly walk through the different things I have on here. So this is our 2 different cell lines and our in vitro expression, sort of split by the different formulations that we've made. And you can see we've tested multiple times. So each individual series here is basically a different well and plate. So it's the same target formulation, just a couple of different replicates. And then you can quickly scroll through that. I have LNP size. So this is just the diameter. It's a very large bar chart. Box plot of the size. And similarly, I have for encapsulation here. Usually want things that are higher, like closer to 90% or more. And then finally, I have, this is more on the input side. So this is just a representation of the ratios of the different components that we put together as a bar chart. I have the component distribution. So based on the 6 different components here, this is basically the mole ratios that we've explored. And then finally, this one didn't turn out. This is just a plot of the different components. So now the fun begins. So I can go to this and basically say, I want to see only the ones that have this component. I can scroll through here and say, these actually look pretty good. They're performing quite well. And then I can go look and say, well, is there something interesting about the sizes? I can blow this up, maybe. Yeah. So actually they're all, roughly around this 120 to 160. They're also very tight. So I can see there's not a lot of variability in the sizes. And similarly, you can do the same for encapsulation. The other nice thing is it shows you highlighted which ones these are. So you can kind of see what's been, what formulations have been explored. And if you hover over them, you can actually see, I don't know, the particular box here highlights up for the sizes and that. So it's a very quick way to explore this high dimensional data. And then here, I also like to play with this and say, okay, I only want to look at the ones that have low percentages. So I'll just highlight these and go back. And so now it only shows me that set. And so one of the questions I ask is how do our scientists interact with this very large high dimensional data? And to me, this is the way, and this is how we're approaching it. And I think about it a lot as learning to swim. I could give you a lecture on how to swim, and it's basically meaningless until you get in the water and start moving your arms and feeling how the water pushes back. I think it's the same way with some of these data explorations. So, real quick, does anyone want to see a correlation on the fly? What would you like to see?
We do size by NANCAR cell type readout.
So you can get close. You can do size, average. And this on the left I already had is expression. So this is a quick way, management asks a question. It's well, what does it look like? So you can see, the best performers have sizes in this range. So you could say, all right, well, we should only look in that range, but I'm more of an exploratory person, and I look at this white space here, and I look at some of the white space here. I was like, well, did we really explore the space if there's all this white space over here? I was like, why can't we go there and see what's there? So yeah, and similarly, you can very quickly switch to, like say, encapsulation, efficiency, and same sort of thing. You could say, okay, this is the box that I want to be in, but also like what happens when it's very low. So you can get a distribution and get a sense for how these things are working. All right, I'm going to digress, sort of step back and ask, like, how do we get there? And I think it's really this data first approach, and we spent a lot of time thinking about what data is, what it means, what information is, and again, like working on the sort of philosophical underpinnings of this. So one thing we really focused on was capturing data and not information. So I'm going to digress a little bit and say, what is the difference between data and information? So data is something that doesn't really change over time and can be used to support different hypotheses, different worldviews, and that sort of thing. And I think the best example of this is basically the positions of planets and stars in the sky. That data goes back thousands of years, and that data is good for fitting a sort of geocentric model where you have epicycles, and it actually works reasonably well for planet position in the sky. But then you can also use the same data to fit a heliocentric model. So that data is that is like to me the gold standard of how we want our data to be. We want our data to be useful regardless of our scientific interpretation or how that is evolving. And so these are sort of the definitions we've been working with. Is like data is very factual. It's like what, where, when, who, how. They're definitions that I think do not change over time, or if they do change over time, it's very, very slow. An example of that would be a timestamp. Information are all things that are derived from data, and they have built-in assumptions, and they require either a worldview or a scientific model of the world. An example is morning. So if your experiment goes wrong at 3 a.m., is that in the morning or is that overnight? That really depends on your perspective, like how do you consider 3 in the morning? So I'm going to play a little game here quickly. Is it data or information? And the cheat code that we always use is if we're arguing over it, it's probably information. And so we want to put it in the information bucket. So we're going to do your left hand if it's data and right hand if it's information. All right, so the lot number of salt used, is that data or information? All right, lots of data. It is data. Percent purity by HPLC, data, information. Ooh, interesting. I have different hands up. I actually think that's information. Because you have to have a view of what purity means, and you're assuming that your HPLC is catching all of those impurities. So that is something that may change over time as we learn more about the process and other impurities that may come into play. So UV detector intensity of the HPLC, is that data or information? So this is data. And this is what we want to actually store, because if we store that over time, we can actually change our interpretation and the information content later on. But if we only store that purity value, we can't go back, or if we didn't store the data piece. LNP particle size, data or information? Cool, I like it. I've got some information in there, yeah. It requires a lot of assumptions, and how you measure it, it's usually a distribution that you get from dynamic light scattering, a whole bunch of assumptions that go into those. Mass of salt added, data or information? That one's data. Concentration of sodium ions. So I think this is information, because it requires a chemist-like view of the world. And if you think about salt being dissolved in water, is it Is it fully dissolved? Is it partially? Like, how do you account for like all of those sodiums? Lipid to RNA ratio. I missed that one. Yeah. Again, sort of a default rule of thumb is like, if it's a ratio, you're implying a worldview on it. Like, that's information. Location on a liquid handler deck. Okay, I think that's data. Stability time. Yeah, information. A lot of assumptions go into that. Like, do you count like the time it took you to get from one place to another? Flow rate of addition, like if you're adding material one to the other. I actually put this on purple. I think it depends. It depends on how you're measuring it. If you have a flow rate sensor, then it's data. But if you're calculating it based on like start and end time and assuming you added it at a constant rate, it's probably information. All right, so why are we doing all this? And really, this is like to get that data foundation that enables us to build up to all the things you want to do. So we want to be able to take that data and have this information layer on top of it, where we do some interpretation of the data. Then going up further, we want to get the modeling and predicting. And so this is where that key data foundation and the contextualization that happens in the information is really what's going to power like our AI modeling. We're doing a time, 20 minutes, yeah. So again, like going back to the example I showed earlier, it's like really splitting the make and the measure parts. So the measurement stuff is actually fairly easy because like once you get a sample to an instrument, like you know what the sample is, as long as you have a unique ID to label it. The only challenging part is capturing all of the preparation that go to that. So that's like pretty straightforward. The sort of making part is much more difficult to make modular. You can have different inputs at different time. You can have waste where you measure, but we want to change those and change the order of addition. And what we found is it's better to keep things as discrete chunks and basically use sort of a graph to enable sort of a rigorous data capture, but then stitch it together as a more like something that looks like a process flow diagram later. And this is, by doing this, we're getting to a point where we can pull a lot of this information automatically from the logs of the instruments that are doing it, or if they're human beings doing it, the sort of the path that they took. So just to give you a highlight of how quickly this has enabled us to change, we built a pilot system for making and measuring LNPs. And at the end of 2023, it was basically just a liquid handler and a few instrumentations and some basically some racks of consumables that we can use. And then we moved buildings and rebuilt the thing to a larger system, added 2 more robot arms, more instrumentation, and another liquid handler. And so this is basically what that looked like in the old building on the left, and on the right is in April, added more capacity, more capability. Again, this is just to show again the picture of the pilot system. So this is the liquid handler we used to either prep plates or make LNPs, and behind it is a rail there's 2 robotic arms that sort of move it between them. On the right, we have some instruments here, we have some open space. And because this is our pilot system, we're basically testing, like pulling things on and off instruments, both physically and also on our data model. And finally, this is like from the other way. You can see on the right our racks for tips and plates and all those things that we need to use in order to fuel this system. And so by doing this, we actually got a lot of learnings that we use to improve the system that we're building, so our V0 system. One thing we very quickly noticed is that dynamic deck building is hard if you're just using the liquid handler to load and unload the decks, and we're spending more time doing that than actually making LNPs. So we immediately changed how we were doing things and actually moved to a Mitsubishi industrial robot that all it does is build and tear down decks. And then we slide those in on a conveyor belt so that the liquid handler is doing what it's good at, which is handling liquids and not building and tearing down decks. So that was a very quick pivot that we were able to make. And the other thing is like the data entry part is difficult. So we're trying to capture really everything about the materials going onto the system. And we need to do it in a way that doesn't have our scientists at the computer doing data entry like day in and day out. So that's something that we've been working on, is like, what's the quickest way we can go from material onboarding to it's digitally registered in the system, and then we like never have to think about it. And then I will say the material and process data capture is still challenging. We're still building that. But I do think we have a vision to where we can get the capture right and do queries on the data afterwards that give us a full tracking of what the material composition was over time. Now I have some fun pictures, so finally getting the robots. So this is a picture from PAA, who's our integrator, building the two of the three work cells. So one is focused on making LNPs, the other one's focused on characterizing them. And the third work cell is basically storage. So I'm gonna play this. So this is just to give you a sense of like the size of the system. On the left here, you can see the robot arm that builds the decks. This is the analytical cell on the left. On the right, which wasn't complete at the time, is the making the LNP cells. And then I'll play this one as well. This is just a video of the robot arm actually reaching and grabbing an already built deck. And it will rotate it down and around and load it onto the liquid handle. It will slide over into place. And so this, you can also see around the arm, has places to unload the deck, throw away the trash, split all of that. So there's really a lot of logistics that we have put into this, and we're really thinking about trying to do it in a way that enables us to get like up to 2,000 LNPs a day and run it with a reasonably small crew. So that is our V0 system. And then basically, we took some of the data that we have from our pilot system, and this is really the value slide for me, is we did a comparison of how do we do things manually, how do we do things on a pilot system, and like what is our projections for the V0. And not only does it save time, like we're basically able to do years worth of work in weeks. So this was like a stability study that took 4 weeks. So I can't cheat time. A 4-week stability study is always going to take 4 weeks. But other than that, it was like, it can go like very quickly. And the big thing for me is like the cost per sample. So we're actually able to reduce that through this process by miniaturizing some of the things and automating it. But really the cost per data point just plummet, because you're getting all of this data and all this contextualization in a much more automated fashion, and you're not asking a scientist to go fill out ELN after ELN after ELN of all the context. And so by having robots do some of this, so you can just really get so much more data for free, and that's really what we want to power everything. Okay, so tools we leverage to drive cultural change. I kind of showed one already, which is Spotfire, so data visualization exploration. I think that's one that's been approachable for our scientists and people are comfortable using. We are definitely going to go deeper into the data analysis, and we have our own computation teams working on AI models and statistical analysis that you can take that data and do more with. The others are, I'll talk about Data IQ next, which is, it really enables us to flexibly stitch together the data and add that information context. And so this is, to me, this is like really important for enabling scientists to sort of test their hypotheses on that different data set. So this is where you build up that next layer. And then artificial, which we've been using, they're sort of like the highest level orchestration for both driving our instruments. So we have a couple of systems that we're already basically commonplace using them to drive some workhorse analytics. And then they're also packaging up and contextualizing all that data and setting it to the right places for us so we can start to build this data set to utilize for AI. So one of the, I guess the key things for driving adoption by science is like really enabling them to collect these massive data sets and then like demonstrating and sharing the value like with the scientists. So this rapid end-to-end data flows and really empowering them, I think goes a long way to getting them to do work differently. And they see the value. So my team in particular we look for those pain points that the scientists have and try to address them directly with these types of systems. And then, deploying flexible standards. So this is a big thing for research is you need to standardize some things, but you need them to be flexible enough to capture the things that you're doing. So this is always something that we're iterating on. And then finally, really empowering scientists to make changes and play. We're researchers. We're, on the frontier of what we're trying to figure out. We are going to do things different all the time. So this is just a shot of one of our canvases from Data IQ. And so this is where we take the data from all these different sources. We do some manipulations and basically build the tables that then power our Spotfire. So a lot of the calculations and manipulations that happen here. And we've had success building these for this project and other projects and handing them off to some of our more junior scientists. So some of our senior research associates that are only a few years out of school, we've built some of these, handed them over, and they're actually doing the changes and the maintenance themselves. So this is something that, it's difficult to build digital tools and then maintain them. Doing it using something like Data IQ, we can actually empower the operators to do a lot of that first line maintenance themselves, and they'll come ask my team or some of the other support from Data IQ that we get to do things on a consulting basis. But a lot of the day-to-day use and maintenance is driven at that operator level. And then artificial. So really having artificial do 4 things for us. One is the sort of lab digitizations, which I don't know if you saw David's talk or visited the booth. This is basically a digital twin of all of the things that are in our system and how they operate. We're also using them for digital assistance, so that's helping humans, which I think we got the term from them, sneaker bots is what we're calling them, do things in a way that's captured just as if a robot would do it. And so that's really building the data and the context. And then we can do workflow design, and we're also using them for orchestrations of both driving these systems or complex workflows and that are both hybrid, human-driven and automated. So this is actually, I think, an early version of the pilot system. So this is what the digital twin looks like. It has all the different equipment on there. If we were, when we went to rebuild the pilot system, I wish we had been farther along in this journey with artificial, because we probably would have done the rebuild in the digital space before we did in the physical space. That's where we're going. We have all these things now. The rebuild of the pilot system will probably happen first a couple of times digitally before it happens physically. And so we've always taken the view of that system should not look the way it does today 3 years from now. It's if it does, then it failed. All the instruments can be pulled out and swapped. So we want to use the digital twin to test out concepts before we actually implement them in the real world. We also use them for some of the workflows that operators do. So it highlights sort of the key activities that you need to do while running the system, and sort of captures that who, what, when type data. We've already put these into place for a couple of the key assays that our formulation scientists do on a routine basis. And so now if you want to go get size on a plate, you actually go run it through artificial. And all that data is being captured. They get the data they want to report out, but we're building that database, and it's no extra work on our operators. And so this is just another example of how the assistants can walk through step by step and show people go get this thing from this place, move it here. What does it look like when you're adding a plate or taking a plate away? All right, and lastly, again, building on some things that David was saying in his talk, we're using artificial to package up all our data and the context for how that data got generated. So really building the data sets that AI need to drive things forward for us. Okay, I'm getting to the end here. Coming back to what drives value for patients. So I hope I've shown you that really it's getting to approvals is all about data and how fast can we do the data, how good is the data. I showed sort of an example of that. And then the question is, okay, if we want all this data, how do we get it? Automating it with robots, getting that higher throughput, higher cycle times. And then eventually, like, I really do think that we're going to get to a point where a lot of the questions that scientists are asking, both on design and making and measuring, you're actually going to be asking it in silico. You're going to be looking at a data set and you're just going to be querying the data. It's like, have we even looked at this before? It's like, I want an LMP that goes to the toe is a example that one of my colleagues uses is like, okay, what do we have in our inventory that we've already looked at that goes close to the toe? And it's like, here's a suggested space, and then you start sort of iterating from there. So I do think like this is where we're headed. And quickly, I want to thank like two of our, let's say, visionary drivers of the work. So Russell Umstead was actually a scientist when he started pitching this crazy idea, and then working the room of different collaborators to sort of get this off the ground. And Andrew Coates is one of our engineers that really helped build the team, build the integrations, and really recruit the diverse skill sets that sort of like make up this team. And I also want to thank Carrie Benanato, who was our VP at the time, for like taking the risk to sort of see this through. She's actually the chemist who synthesized the aminolipid that is in our vaccine. So very, very important person. With that, I want to thank you for your attention. I'll definitely take some more questions. Leave the acknowledgement slide up. Thank you.